Right, here we go. 2 teaspoons of Botox is enough to kill
I know
the
this,
entire
entirety of the
United
UK.
Kingdom. Yeah, the entire of Britain. That was the one I knew you knew.
Oh,
But
right, okay. We discussed
2
this.
teaspoons
Is this
is
the one
enough
you
to
said we've
kill.
discussed?
Yeah.
It's the most toxic chemical to humans.
Guess what the lethal dose
Because it's a
is?
really potent nerve agent, right?
Yeah. Guess what the lethal dose is?
The
Roughly.
lethal dose.
Yeah.
Is this a number of molecules?
I'll accept a range. It's in a weight measurement. Ends in
Right, okay.
grams. It's something grams.
Okay.
You can guess in standard form the number
I'm
of
going to
grams.
say 1 times 10 to the -7 grams.
You're roughly 5 orders of magnitude out.
What, 1 times 10 to the -2?
No, 12.
1 times 10 to the -12.
It's 1.3 to 2.1 nanograms, which is 10 to the -12, I believe.
Kilograms. 10 to the
Kilograms.
-12 kilos. I said
Yeah.
grams. Wait, so I was, because I was doing -7, which means I was in the hundreds of nanograms, Sam. I wasn't that bad. A nanogram is 10 to the nano is 10 to the -9.
Oh, right. Oh, well, shut up. Who cares?
I did 0.1 micrograms, which means I'm 100 times what you're doing. God, I wasted a lot of time there. Yeah, sorry. Is that your fun fact?
I mean, my fun fact was expanding on why botulinum toxin is so damn toxic.
Okay.
Do you want that expansion, Henry? I mean, what do you know about the mechanism of action of Botox?
Is it to do with the myelin sheath, Sam?
No.
No. Okay. Is it to do? It's to do with synapses. Is what it is.
It is to do with synapses.
It's going to block a neurotransmitter in a synapse.
In a way, no.
Is it filling a synapse with a neurotrans? What is it doing? It's
filling it. Well, I guess it does block it, but not in the way that it feels like you're implying. Essentially, there are these things called snare proteins.
Right.
Right. So when you do a synapse is a gap between 2 nerves, and essentially what happens is you release a vesicle of neurotransmitter that goes between the presynaptic neuron and then goes into the postsynaptic neuron, and then it transports neurotransmitter between them. Now, what you need to happen for that to work is you need the vesicle at one end of the synapse, at the top end. You need it to like fuse with the wall, fuse with the membrane, and as it fuses, it releases the stuff it has inside of it. Right? That's called exocytosis. That's a very important thing.
Wait, so you've got your nerve, you've got the little stringy bits that come off the end of your nerve for your connections.
Yeah, and at the ends of those stringy bits, you have a little wall called the synapses. In the synapse, in the cell, there are do you know what a vesicle is?
It's like a hole. It's got this little compartment.
In a way, it's the opposite of a hole.
It's a little box.
Yeah, basically. It's like a sort of box of cell. Like imagine it's like a bubble of cell membrane inside the cell.
Right, and it's got to go flat against another surface in order to explode out the neurotransmitter, which is a chemical inside of the box.
So the synapse is between the 2 cells, right? That's the gap.
Yeah.
And then the vesicle is in the first cell. It hasn't been released yet. Okay, here we go. So imagine the synapse is a street. The synapse is like a road, right? And there's one house on one side of the road and one house on the other side of the road. You haven't left the first house yet. So there's the water balloon is in the first house trying to get through the wall. Once it's got through the wall, it's in the road, and you just release all the neurotransmitter loose in the road, and it just lives in the road, and the concentration goes across to the other house, and the other house on the other side of the road sucks some of that up and then takes it into the house.
Why is there a wall in the middle?
Well, that's just the wall of the cell. You wouldn't have a cell if there wasn't a wall.
Oh, so it's a cell wall.
Well, it's a cell membrane. I shouldn't say cell wall. This is all about leaving the first house. So your exocytosis on the first cell. You're trying to get the neurotransmitter out of the first cell to begin with into the synapse. Your goal is to get to the synapse, cool. Is the idea.
Right.
And it's all very useful. But the thing is, activating the synapse. You want this to happen in fragments of seconds. You know, you don't have time for your balloon to saunter over to your brick wall, right? So snare proteins help with that. Snare proteins essentially keep the vesicle, which already has all the neurotransmitter in it. It keeps the vesicle right next to the cell membrane. It keeps the balloon right next to the wall, as you will. There are 3 snare proteins involved in this. Okay, 3. There's one called syntaxin. There's one called SNAP25, and there's another one called synaptobrevin. So you got synaptobrevin, which is on the vesicle, right, on the balloon. It's sort of just sitting there. And then syntaxin and SNAP25, they're on the wall. They're on the cell wall. And then what they do is essentially you get a little sandwich, basically, where it's synaptobrevin, SNAP25, and then syntaxin all stacked together in that order in a little sandwich, and they bond together nicely in that way. And because they're all bonded together when they're in that little sandwich position, that keeps the vesicle on the wall, right, ready to go, right against the wall. And that's called, you form something called a core complex.
Wait, so it's like a sticky mechanism.
Yeah. It's cis-trans bonding between the 3 proteins that are attached to the vesicle and to the wall, and they all slot together. So now you got the vesicle on the wall of the membrane. It's all well and good. Loads of these form core complexes, which is what this is called. You get loads of them building up on the wall of the presynaptic neuron. And then when you got an action potential coming, when you got a neuron being activated, calcium comes into the cell, because that's what happens. You get calcium gates opening, calcium rushes in, and there's another protein I didn't tell you about, Henry, called synaptotagmin. That's not even in this complex, but it's just stuck on the vesicle. So the vesicle has synaptobrevin and synaptotagmin. And what synaptotagmin does is it waits for calcium. And then on the CA2 site, I think, of this protein, calcium binds, and that essentially triggers the core complex to, I don't want to say contract, but it does a similar thing to contracting, where it smushes the vesicle into the wall. So usually that would sort of happen passively, but then once these 3 proteins detect the calcium and they conformationally change, and the vesicle just gets smashed into the wall, you're essentially just pushing the water balloon into the wall.
Great.
Everything comes out, and that all happens at once because it's right there. So it's all very quick, very coordinated, all at once, because they're all in the same place. You don't have to wait for them to fly over. They're already there. But with Botox, so loads of different kinds of Botox. You got types A, B, C, D, E, F, G, whatever, loads of different kinds. Each of them attacks different snare proteins, but all of them attack snare proteins. So, like, for example, what they do is they basically just cleave them. They just get in and they just cut the protein into, and they stop it from being able to properly form. So you can't make the snare proteins. So you can't form the core complexes. So the balloon can't be smushed into the wall. So when the calcium comes, who cares? The balloon's just sitting there. It's not even near the wall. It's nowhere near where it needs to be.
Which means you're not triggering another cell, which means you're not triggering any more calcium.
Which means you're not releasing any, you're not releasing any neurotransmitter. The acetylcholine isn't leaving. It's not going into the next neuron, and then the next neuron doesn't care. Nothing happens. So it's paralysis.
You're breaking the chain of neurons at one point, and then the message can't be transferred at all.
Essentially, yeah. And you're breaking the chain of all the neurons around there. And so Botox A, that's how it gets SNAP25. But that's the most toxic one, I think. And that's at least the one used most often. But the rest of them are also very bad, and they attack stuff like syntaxin and synaptobrevin and whatever. And then once your snare proteins don't work, essentially you don't work. Instant paralysis, basically. No chance whatsoever. And yeah, I mean, 2 teaspoons will kill the entire country. Is pretty fucking cool, I thought.
10 to the minus 9 grams.
10 to the minus 9 grams, 10 to the minus 12 kilograms.
Yeah. Jeez.
Nanograms. This is 1 to 2 of those. So, okay, great. There you go. So that's the bit of Botox for you. And to think we inject that into our face willy-nilly.
Yeah. How much do we inject into our face?
A decent amount, but the thing is, it doesn't get into you direct it directly into your blood. So, sorry. You direct it directly into your muscles. So those muscles are dead, which is why they're all paralyzed. You're paralyzing your muscles when you put Botox in. But the thing is, it's not going anywhere else because it's that bad. Like, it's so bad that it just stops with your muscles. But it's not getting into your bloodstream because it's so targeted.
If it got into your bloodstream, surely... Ooh, okay. So it's not going to do anything to your blood. Right. And then it diffuses out of your bloodstream at different points around your body, let's say.
Yeah.
And the fatal part would be stopping your heart, or making you a bit brain dead.
Thing is, it does depend. The ability to spread from the injection site is what kills you, and even if a tiny bit of it does, it will just it will paralyze your heart or any part.
So you need to somehow contain.
Basically, it paralyzes any muscle that it happens to be in, and if that muscle happens to be your heart or around your brain or whatever, then you're dead.
The lethal dose, when applied directly to the heart at the key point in the heart, can be as small as X.
Yeah, essentially.
Is what the fact is.
The lethal dose is defined in all sorts of ways, so it's it's toxic in very many places.
All right,
And
Sam.
it causes
Thank you for that.
spasms as well, which is not good.
I learned quite a lot there.
Yeah, Botox.
That's a piece of knowledge that I'm grateful that I have now.
Each individual ant is kind of very stupid.
No, actually, Sam, I just need the toilet.
Oh. Pineapple eats you.
The physics question is everything just balls? Or is there more to it?
Why is he that? Why is he that?
Oh, I'm getting a bit warmer these days. Maybe I should, disperse my ovaries into the world.
Mm-hmm. I do that, yeah. This is non-falsifiable. This is not science. I'm not a fan of that, Henry. I'm gonna be honest.
Guess I'll wait for the sun to blow up.
Why are you still googling Fast and Furious, Henry?
There's John Malkovich as Snail.
And then this presenter was like, "Can you maybe elaborate on that? Maybe just for the viewers at home, give a layman's explanation." And I was like, "I can't. No, I can't." And then it goes down, burn out. That's ugh.